Okay, on to lecture 15C. We're going to only lightly touch on what's there in the textbook. In fact, let me show you that right now. Oh, you share. Okay, there it is. What we're looking at is the processing of the polypeptide proinsulin, which is one of the steps that has to happen after translation. And now I'm going to go back to my original share whiteboard and remind us about what that means. Okay, post-translational control. We started off with a gene. That's basically DNA, right? And not only is it DNA, but it also has to be associated with that, non-methylated DNA. The cytosine residues and sometimes carry methyl groups, which will give you a coarse level of control, causing the DNA to be generally turned off. Gene activity is going to be really low when you have methylated cytosines. Preferably, the activity is going to be heightened if you have histones in that same area acetylated. Okay. And we call this the coarse level control, kind of like coarse focus knob on a microscope. Coarse control, because it's kind of an it's not an all or none thing, but basically it can take out large blocks of chromatin from activity, or activate large blocks of chromatin from activity. Okay? So above and beyond the coarse level of control, we also had the fine level of control, which was based on whether or not we were initiating transcription, right? So transcriptional control level 2, or the fine level of control, call it fine control. Because we're actually putting some detail on which genes are being turned on, which genes are being turned off, was happening through the activation of the RNA polymerase, right? So RNA polymerase is brought in with transcription factors. They have to be present. We have to have the right combination of transcription factors. We have to have RNA polymerase. Coming in, and if the gene is turned on, we get through through transcription. What we end up with is an RNA transcript. Okay. And we're not at the level of translation yet, right? I mean, after RNA transcript is formed, once we actually have the copy of what's there in the RNA, what's there in the DNA, right? The RNA transcript then has to undergo editing, right? The editing includes removal of introns, removed. Remember that? Gave that to you. Previous video, we're going to be stitching together the exons, and we have to add a GTP cap and a poly A tail in order to make the RNA complete. To complete the messenger RNA molecule, the mRNA, we have to undergo all of those steps. Okay, and the mRNA then gets exported from the nucleus. Into the cytosol, and that's where we have the process of translation. Okay, so we have attachment by the ribosome. Attaches, we have to target the right location. And so in this case, we're talking about two hormones. Both hormones are, these in particular, are polypeptide hormones. They're so basically proteins, right? And so it's, they're ideal for us right now because they're giving us a chance to talk about gene activity. And since they are polypeptides, you know they're going to be coded for by a gene. And so there's going to be an insulin gene, there's going to be an angiotensin gene, and the final gene product is going to be this polypeptide hormone that are going to have physiological effects, hormones. Yeah, and what's important about here, a hormone is going to be secreted, right? Which means it's going to have to go through the endomembrane system. And so if you recall back to other parts of your BIO110 semester so far, you know that if a polypeptide is going to be secreted, it has to be produced within the endomembrane system, which basically means it's going to go to the rough ER. Right? Remember, there are free ribosomes that are going to be translating and producing proteins that stay inside of the cytosol. And then you've got the rough ER, in which the roughness, the specks on the rough ER are basically the ribosomes that are secreting, right? I'm sorry, not secreting, the ribosomes that are producing the polypeptides in which the gene product is ultimately going to be secreted from the cell. Because from the rough ER, it goes to the smooth ER. From the smooth ER, it goes to the Golgi. And the Golgi produce vesicles. And the vesicles eventually make their way out to the outer membrane, out to the plasma membrane, and we have secretion into the bloodstream, into the digestive tract. Basically, wherever the hormones are released, they're going to be released from vesicles produced by the cells that are going to be secreting the hormones, right? And so, coming back to our story over here, when they're exported from the nucleus, they've got to be targeted to the attached ribosomes on the rough ER. And only when it's attached to the rough ER and we're now producing the polypeptide inside of the lumen, that's the empty space inside of the rough ER, are we actually going to have a polypeptide. Okay. And that's the process of translation. Okay. And coming back to our general story here, okay, we know that we've got control. I'm just going to mark off the things we've talked about, right? We've got control of gene activity happening at the coarse level, where we're just turning on or turning off broad swaths of chromatin. Okay. Then we've got the fine level of control, where we're actually precisely turning on or turning off a gene by using transcription factors and creating those little shepherd's crook bends, and that attracts the RNA polymerase. And so, these two are the we call these transcriptional control. Of gene activity. Because they're really determining whether or not the gene ever makes it to the process of RNA. The information is the information actually copied into RNA or not. And so that's where that's one form of control, right? And after you have the RNA transcript, I'm going to switch colors here, move over to maybe a green, right? Once we have the RNA transcript, there are more things we could do. Maybe not necessarily control, but we can alter, for example, by using different patterns of intron removal. We can produce different versions of a gene product. And so, yeah, we talked about these two. And this is all important stuff that goes on in the editing of the RNA to make the RNA molecule. And then once we get to the RNA, we can do a couple of things. We can send it to the rough ER like we're doing here, or we might actually send it to, we might not have the appropriate signal to attach it to the rough ER, in which case the protein would be made into the cytoplasm, right? We're not doing that here because we're talking about secreted polypeptides. And now that we have the polypeptide, there are things that can happen after the polypeptide is done before we actually get to a final gene product. Okay. Which in our case right now is the hormone. Okay? And so it's actually possible to um, to elevate or to attenuate the activity of the final processing, call it processing of the final gene product that's occurring in the rough ER, in the smooth ER, in the Golgi apparatus. There are a lot of steps that are required before you get to the final gene product. And you could either accelerate that or you could turn it down, slow it down. Right? And so here is kind of like what we're talking about in this video, in this lecture. And the two examples we have are insulin, which is an enzyme, which is a hormone that you're probably aware of. It's the hormone that brings glucose out of the bloodstream and takes glucose out of the bloodstream and stores it in liver glycogen, right? And so when you eat a large meal, your blood sugar goes up. You need to release a greater amount of insulin, and that's basically what's going on, right? And so how do you make that final version of insulin? Well, in the process of making insulin, I've got a slide here for that. Okay? This is basically a reproduction of that thing that we saw in the textbook. We're starting off with an inactive version, proinsulin. Okay? This is kind of like the version, the whole polypeptide is A, C, and B. Okay? There's a reason why C is in the middle. Talk about that later. But the inactive version is a long polypeptide that in order for it to reach the final stage, insulin, we've got to do a couple of things. Okay, we've got to fold it around. We've got to bend it around like a paper clip and attach the B polypeptide to the A polypeptide through a couple of disulfide bridges. You might remember that from a first unit where we talked about the fact that it can have cysteine residues forming disulfide bridges, which stabilize the protein shape, right? But in any case, we've got a couple of positions for the, this is going to attach to this, actually here, and this cysteine is going to attach there. Actually, a couple more that I'm not showing you, but get the idea. In order to get these things around, we've got to fold it around, like I said, like a paper clip, so you get this structure. And now we can actually make those disulfide bridges. Right? These disulfide bridges are going to hook up. And then the last thing we do is to use a protease, and we snip off this C peptide, which then becomes kind of like cellular trash. Right? No, we don't need it. It's just the thing, the part of the paperclip that has to fold around in order for us to make those final bridges. And the final insulin version will be this kind of, it's really a broken polypeptide. We buttoned it together in two spots, and then we actually split it. We took out a middle portion. And the insulin in its final stage, this is the active version. And what this allows us to do is we can actually store proinsulin in an inactive form, and then just do the final activation, the conversion of proinsulin into insulin at the very last stage when we actually need to release the insulin. Okay? And so that's basically what happens. If we were to interrupt the conversion of proinsulin to insulin, that would basically make insulin less available to us. That would not be a good thing. It's actually not an avenue that I want to follow, but because I have a better example of why we would need to potentially use post-translational control in a particular situation. And so the situation we'll be discussing is one that literally is the difference between life and death. It's a life and death situation. If you were to sustain a severe injury and maybe have a substantial amount of blood loss, your body is going into shock. And shock is basically extremely low blood pressure. And it's the thing that basically causes death in the case of blood loss. The drop in blood pressure is not something, it's a really bad thing, right? And basically the long story short version of this is that when you are going into shock, the thing that you need the most is to elevate your blood pressure, right? So elevation of blood pressure is what we need, okay? And one of the ways that we can do this is to have the hormone angiotensin 2, which will induce your arterial system to vasoconstrict, right? So vasoconstriction means that we're narrowing the blood vessels leading away from the heart, which reduces the volume. And by reducing the volume, it increases the pressure. And that's something you'll learn about in physiology. Basically, reduce volume, increase pressure. Okay, and so it would be a really good idea for us to have a ready supply of angiotensin 2 ready to go at any given time. And that's exactly what's happening. Okay, and so the idea here is that in your cells, we don't stockpile angiotensin 2 because we don't want the vasoconstriction all the time. But we need to be able to initiate that vasoconstriction on a moment's notice. And so the transcriptional control mechanism that we looked at in the first slide, this idea of, okay, let's turn on the gene for angiotensin 2, and then we're going to activate with transcription factors the RNA polymerase, which is then going to give us an RNA transcript. And then we've got to edit the RNA transcript and export from the nucleus and then send it over to the rough ER, translate the angiotensin 2 messenger RNA molecule, then process it, and finally get the gene product. I mean, to go through that whole process would be incredibly slow and you'd be dead. It wouldn't work, right? And so in this particular case, you can actually see why it's a good idea to use the post-translational control on whether or not angiotensin 2 is there and available or not. Because if we store the inactive version, angiotensin 1, okay, inactive, but abundant, as like stored source of angiotensin 2, then if we need to have a vasoconstriction because we're losing blood pressure quickly, all we need to do is to convert the angiotensin 1 into the angiotensin 2, right? And I am taking this someplace that I think is relevant for you because the enzyme that converts angiotensin 1 into angiotensin 2 is named appropriately enough angiotensin converting enzyme, or ACE for short. Okay. So yeah, I mean, when we need the increase in blood pressure, we kick up the angiotensin converting enzyme, we activate the angiotensin converting enzyme, it converts the angiotensin 1 and angiotensin 2, and right away we have the final gene product that we need to result the effect of what we need to have happen. Okay. So so that's pretty much the end of our story. If you have to ask the question, well, why post-translational control? Because after all, we've got so many great and wonderful ways of controlling gene activity with transcriptional control. What would be a situation when post-translational control, in other words, we go through the entire process of polypeptide synthesis. All we're doing is waiting to put the final touch on the polypeptide. And now we've got the angiotensin 2. Well, why would we need to do that? Well, in the case of angiotensin conversion, we might need to have that gene product a lot faster than what you could get going through the process, starting with transcriptional control. Okay. And I hope that is very sensible it's very straightforward. And the answer to this question is, I think, pretty obvious. Uh, Maybe not obvious if you don't have a chance to think about it, but think about it for a little bit, and it should be obvious to you. Okay. Now, the thing about this particular situation is that this angiotensin converting enzyme is also a target for drugs whose intention is to control blood pressure, because, as you know, hypertension, high blood pressure is a major problem in at least the United States population, right? And so if we have hypertension drugs, if we have hypertension, your physician would probably be prescribing some type of hypertension drugs, which might be a beta blocker, which can slow the heart rate. If you slow the heart rate, that would cause you to not have as much pressure in your cardiovascular system. You might get diuretics, which would cause you to pee a lot. And if you pee more than you otherwise would, that reduces the volume of the blood. If you reduce the volume, of the blood, okay, don't get confused with this, right? Here we're talking about reducing the volume of the blood vessels, right? If you have the same volume of blood vessels in a reduced volume, if you have the same volume of blood, okay, stop, rewind. Okay. If you have the same volume of blood in a smaller volume of blood vessel, you're going to have increased pressure. Okay, but if you have the same volume of blood vessel holding on to reduced volume of blood, that's going to lower the pressure. Right? And so, yeah, if you take a diuretic and you reduce the volume of your blood, that's going to have the effect of lowering your blood pressure. Okay? And the third major class, and these are the Opril drugs, or Ipril drugs that tend to end in Opril or Ipril, like lisinopril, right? These are in the class of ACE inhibitors. And ACE inhibitors basically do what you would expect them to do, right? If you got this enzyme, you could inhibit. Remember we talked about enzyme inhibition? You do something to this that would make the ACE unable to convert angiotensin 1 into angiotensin 2. And yeah, to the extent that people with hypertension could benefit by reducing the total amount of angiotensin 2 that's there in their system, right? By inhibiting the ACE, will be reducing the angiotensin 2. You would have not as narrow of a blood vessel. In other words, your blood vessels would be slightly dilated, which would increase the volume of the blood vessels, thereby decreasing the pressure. Okay, the other thing that, and this is a worthwhile thing to note, the other thing that ACE does is it breaks down bradykinins, okay, which causes vasodilation. Okay. And so if you, this is kind of like a whole bunch of negatives. If you reduce the ACE, angiotensin converting enzyme, you'll have less breakdown of bradykinin, which means you'll have more vasodilation, which basically means you'll be decreasing the blood pressure that way as well. Okay. And so, yeah, I mean, I did promise you early on that when we talked about things like enzyme inhibitors, we would be coming back to that story. And I apologize if we've got some negatives and positives to keep track of, but that's kind of like what you need to do, right? If you're going to be a practicing health professional, you need to have some basic understanding of how drugs work and how they interact with the physiology of your future patients. Okay, so that's it for this video. I hope it comes out Final Cut.